Good morning, students. Today we will be discussing about a very important topic called the urea cycle or the ornithine cycle. We know that the liver is the main organ for protein metabolism. It has both anabolic and catabolic functions. In the liver, amino acids are synthesized into large proteins that circulate through the body, performing a huge variety of tasks, including stimulating production of other proteins. Liver also breaks down the protein. For example, the hemoglobin found in the red blood cells, which has expired after 120 days. The liver cleanses the blood by removing the cellular debris and processing excess nitrogen that is produced by chemical reaction within the cells. The excess nitrogen is initially in the form of ammonia. If ammonia is allowed to remain in the blood, it would rapidly become toxic to the body. Hence, the liver converts ammonia to non-toxic urea that is removed from the body via urine. This lost nitrogen is replaced by the food that we consume. All animals need a way to excrete the nitrogenous waste. Most aquatic animals are monotelic. They excrete nitrogenous waste in form of ammonia. The other type of nitrogen excretion is seen in the mammals, where they excrete nitrogenous waste in form of urea. In birds and reptiles, the nitrogenous waste is excreted in form of uric acid. This we have already discussed. The urea cycle is the first metabolic pathway discovered, and this cycle is known as Krebs-Henschel urea cycle, also known as the ornithine cycle, as ornithine is the first member or chemical of the reaction. It is synthesized in liver and transported to kidney for excretion. The sequence of cyclic reaction in which the potentially toxic product of protein catabolism is converted into non-toxic urea cycle is called the urea cycle. It is a cyclical reaction process. The ornithine cycle or urea cycle is a cyclic enzymatic process consisting of consecutive transformation of amino acid ornithine and leading to the synthesis of ammonia. The two nitrogen atoms of urea are derived from two different sources. One from the ammonia and the other directly from the alpha amino group of aspartic acid. The carbon atom is supplied by carbon dioxide. Hence, urea is the end product of protein metabolism or amino acid metabolism. Urea accounts for about 80 to 90% of nitrogen containing substance excreted in urine. Urea synthesis is a 5-step cyclic process using 5 distinct enzymes. The first two enzymes are present in the mitochondria, while the rest are localized in the cytosol. The ornithine cycle consists of the following step. First, conversion of ornithine into citrulline. Second, conversion of citrulline into arginine. And third, splitting of arginine into urea and ornithine. The reaction first and second, that is conversion of ornithine into citrulline and conversion of citrulline into arginine, requires expenditure of energy which is provided in the form of ATP. Reaction 1, that is the conversion of ornithine into citrulline, proceed in 2 steps. A. The formation of carbomyl phosphate, and second, the formation of citrulline. The formation of carbomyl phosphate which has high energy phosphate bond from ammonia, carbon dioxide, and 2 molecules of ATP is required, as is clearly demonstrated in the diagram. Ammonia, which is the nitrogenous waste, is the first product obtained during the protein catabolism. It, along with carbon dioxide, forms a carbomyl phosphate. In this reaction, 2 molecules of ATP is required. Second is the formation of citrulline. In the reaction of carbomyl phosphate with ornithine, the citrulline is produced. The second reaction, where the citrulline is converted into arginine, also occurs in 2 steps. First, the reaction of citrulline with aspartic acid to form arginosuccinate. The reaction proceeds with the participation of ATP with the liberation of adenylic acid or AMP and pyrophosphate. And second step of reaction 2 is the splitting of arginosuccinic acid to yield arginine and fumaric acid. In reaction 3, the arginine is hydrolyzed to urea and ornithine. The urea is excreted via kidney and the ornithine again enters, or rather re-enters, the urea cycle. The ornithine cycle has been found in mammals, frogs, turtles, earthworms, but is absent in snake, birds, and some bony fishes. In this slide, the formation of carbamyl phosphate is demonstrated. Carbon dioxide plus ammonia plus 2 ATP in presence of enzyme carbamyl phosphate synthetase 1. Carbamyl phosphate is formed. The formation of citrulline. This second reaction is also mitochondrial, that is, it occurs in the mitochondria. The steps I have already mentioned. Ornithine plus carbamyl phosphate in presence of enzyme ornithine transcarbamylase, citrulline and inorganic phosphate is formed. Third step, the formation of arginosuccinate. Citrulline condenses with aspartic acid to form arginosuccinate. The enzyme involved is arginosuccinate synthetase. The second amino group of urea is incorporated. It requires ATP and is cleaved into AMP and inorganic pyrophosphate. The formation of arginine or cleavage of arginosuccinate. The enzyme arginosuccinase or arginosuccinate lyase cleaves the arginosuccinate to arginine and fumarate or fumaric acid. The fumaric acid is an intermediate product and it can enter the TCA cycle. Fumarate provides the connecting link between TCA cycle or gluconeogenesis. The fumarate is converted into oxaloacetate. The enzyme involved is fumarase and MDH and transaminated to aspartate. Aspartate is regenerated in this reaction. The arginase cleaves arginine to yield urea and ornithine. Arginase is the enzyme mostly found in the liver, while the other enzymes of urea cycle are present in other tissues also. Arginine synthesis may occur to varying degree in many tissues, but only the liver can ultimately produce urea. Significance of urea cycle. Ammonia is toxic in the blood, hence it is converted into non-toxic urea. The synthesis of semi-essential amino acid arginine. Ornithine is the precursor of proline and polyamines. Polyamines include putrescine, spermidine, and spermine, while polyamines have diverse role in cell growth and cell proliferation also. The urea cycle is regulated as the carbomyl phosphate synthetase is a rate limiting enzyme in urea. It is allosterically activated by N-acetyl glutamate. It is synthesized from glutamate and acetyl-CoA by synthase and degraded by an enzyme called hydrolase. The rate of urea synthesis in liver is correlated with the concentration of N-acetyl glutamate. The first two enzymes in ornithine cycles are present in the mitochondria. Fumarase is also an enzyme of mitochondria, while arginosuccinate lyase is the enzyme present in the cytosol. Urea produced in the liver diffuses and is transported in the blood to kidney and is excreted in the urine. A small